Corona is claiming millions across the globe. The biggest issue which the researchers are facing right now is the ever changing sequence of COVID 19. The strains are making it difficult to identify the new drug. For a drug to be tested in silico, you need to perform docking studies with the protein. And due to this ever changing sequence, the available proteins which are present are also of very little help. Now, you know the amino acid sequence of COVID 19, but you don't know the protein structure. How will you predict the three-dimensional protein structure? Hi, this is Sanket, and today we are going to predict a three-dimensional protein structure for a COVID-19 sequence using Ab-Initio based method. Yeah, so suppose we have a amino acid sequence for COVID-19, and you want to now predict the three-dimensional protein structure for this COVID sequence. So what you do is you take this sequence and then you carry out a blast and of it with your known databases. Um, so what this happens is this sequence is then matched with all the known sequences of the protein, and then finally what happens is a structure is predicted. So this 90% what is obtained actually means that your COVID sequence or this structure what is predicted is 90% identical to some of the proteins which is present in the database. This entire process is what we call as homology modeling, where you're trying to predict where you're trying to predict a three-dimensional protein structure from known structures which is present in the database. To know what actually is homology modeling and what are the different steps in homology modeling, you can go to my video, which is homology modeling tutorial part 1 and part 2, and how the homology modeling is performed using the Swiss model server is being shown. So I recommend that you watch both of these videos and then you can go to how protein prediction using ab initio based methods. So now let's take another criteria into consideration. Suppose we have a COVID sequence, we perform a blast of it with the known databases, and then we predict the three-dimensional structure of the sequence. Okay, and suppose the templates what we are getting for this sequence from the known databases, the identity percentage of those templates, okay, is, let us say, less than 40%. Okay, so we are getting, so you can say that this sequence is just 40% or less than 40% identical to the known PDB structures which is present. Then what? Should we consider that protein structure, and should we still carry forward that protein structure for further analysis? No, because if your identity percentage is uh, 40% or less than 40%, then that protein structure, with the three-dimensional protein structure which has been developed, is completely unacceptable. So what should we do at such time? We move to something called as an ab initio based method. So Ab-Initio modeling conducts a conformational search under the guidance of designed energy functions. The procedure usually generates a number of possible conformations, or what we call as structural decoys, and then your final models are selected. The three important factors which are needed for carrying out a possible ab initio based modeling is the model needs to have an accurate energy function, conformational search, and selection of native like models. So if you ask me what exactly is the difference between homology modeling and ab initio based method is, in homology modeling, you're predicting the three-dimensional structure from known structural databases like PDB. So you're taking PDB structures as the reference, and then based on those reference, you're trying to create a new predictive structure. Whereas in ab initio based methods, this thing does not exist. So what you're trying to do is you're trying to completely determine a new three-dimensional structure now based on some structural decoys and some knowledge of the proteins which is there. So understood what exactly is an ab initio based method. So the tool for ab initio based method is I-Tasser. And today we are going to study, today in this tutorial, we are going to look at how to determine um, a three-dimensional protein structure for a COVID-19 sequence using this ab initio based I-Tasser tool. The link for this tool is present in the description below, so you can click on that link and you can go to the home page of I-Tasser. So let's start the tutorial for 3D structure prediction using ab initio based method. So in order to get the sequence for your COVID-19, if you are a researcher who has a project which is going on COVID-19 and you have the sequences, then it's a wonderful thing, well and good. But if you are a researcher like me who want to work, who want to understand and analyze the different sequences which is there in the database of COVID-19 and try to understand its structure and try to determine its structure, then one such database is your UniProt database. So this is the nucleoprotein of COVID-19 which is present. Now if you go to its sequences, okay. Yeah. So now if you go to its sequences, you can download. So I really don't know whether the PDB structures for nucleoproteins COVID-19 is present or not. But I've considered this as one of an example where we can consider a sequence for your COVID-19. So I download the sequence here. Okay, and just you can see the download faster. So once you click on that, yeah, so this window opens. Now let's go to our tool, the main thing what we want to study today, that is your I-Tasser tool. So this is the link for I-Tasser which has been given in the descriptions. When you click on the link, it brings you to this. So I-Tasser, as I told you, is an ab initio based tool. Okay, so it predicts the three-dimensional structure of the protein based on the ab initio based methods or on the decoys which is there. Now, how do you actually perform this is you take this sequence, okay, you take whatever your unknown sequence, you take whatever your sequence is, you copy it, all right, and then you paste it here. Okay, so your sequence has been pasted here. Now the next thing is you have to run the I-Tasser. Okay, that's how easy it is. But now the only issue with I-Tasser is that you have to fill in the email. So you have to actually create a sort of an account in I-Tasser in order to run this program. Now, if you have an email ID and a password, it's well and good. If no, then if you can see here, click here if you do not have a password. So you click on here. All right, so this is where your server registration comes. So then you have to fill in your name as Sanket, then you fill in last name Bapat, then you fill in your institute, which is MIT College. Now you have to fill in your email ID and your confirmation email ID. Once you do that, then you click on submit. Yeah, once you click on a submit, you wait for like few seconds, and then on your email ID which you have submitted, see to it that the email ID which you have given is correct, because everything, okay, your from further now will be connected to your email ID. So that's why see to it that your email ID is perfect. You will see something called as Yang Zhang Lab, and there it will be coming, thank you for your registration at I-Tasser server. When you click on this, you will find a password. I have actually blurred all of this, okay, but in this section you will find that your password is so and so. So you get now a password for your I-Tasser server. Once your registration is complete, then the next steps are pretty easy. You just have to fill in your email ID, then you have to fill in the password which has come on your email. You can give an ID name for it. It's an optional thing. If you want, you can give, or otherwise it's your choice. Okay. So okay. Once you do that, then you can keep these options as it is. You don't have to go into this. All right. And then you have to click on Run I-Tasser here. Okay. So this is your sequence which you have entered. Okay. And you fill in all the details, and then you click on Run I-Tasser. Now, when you click on Run I-Tasser, for the results to actually come, it can come as quickly as 10 to 15 minutes. Sometimes it can take an hour or so, because what happens is it depends completely on the traffic. Okay, so how much the load is there on the server, based on that, it will take a lot of time. It also has happened that once it took me about 11 hours to generate this thing. I mean, it's not all the time. Okay, mostly the traffic is the least, so you have to wait for max 10-15 minutes for your 3D prediction model to come to you. Now, remember that the results for this, you get it in your email ID. So let's check our email whether we have got the result for our COVID-19 protein structure. Yes, it has come. So if you see here, I-Tasser results for your protein. All right. So when you click on this, yeah. So it says here, this is your job ID, so and so, and here is the link. So these are the various images of the models which has been generated. If you have gone through the homology modeling tutorial, you will know that it generates a number of models. Similarly, I-Tasser also generates n number of models. So now let's click on the link to see the output of our Ab-Initio based method. Yeah, so this is your submitted sequence, which is your FASTA format. If you scroll down below here, it says your predicted secondary structure. All right, that means this is your entire sequence. It is giving us which are the various secondary structures which is present in the sequence. So your C stands for your coil, H stands for your helix here, and S stands for your beta strand. So this gives you the secondary structure information for the sequence. Then if you scroll down, here is your predicted solvent accessibility. So the numbers range from 0 to 9, 0 being the residues are present in the buried surface, and whereas 9 being your the residues are exposed. So this is your solvent accessibility. Then you have some normalized B factors, As you scroll down, it what it actually also does is it tries to carry out sequence alignment with the known PDB structures, and based on that, it gives us the identity. Now, if you remember, your ab initio methods is done only in those conditions if your sequence identity is less than 40%. So this actually holds no value if you have a sequence which is very less. But yeah, if you are dealing with cases such as COVID-19, even that small 30% identity or 20% identity uh, gives you at least some bit of information about that protein. Now let's come to the main thing what we are actually interested in is the five models which is predicted. So these are the five models which has been predicted by I-Tasser. Okay, these are your five models. Okay, now which is the best model, okay, out of these three? Now if you scroll down here, you can see there is C score being mentioned here. So what exactly is this C score or this confidence score? So confidence score is basically for estimating the quality of your predicted model by I-Tasser. The way it is calculated is that it is based on the significance of your threading template alignments and something what we call as your convergence parameters of the structure. What is the range of your C score? Is it ranges from -5 to 2. So where your C score is of higher value, that means it signifies higher confidence. So if we compare the C score of all of these five, okay, we will get to know, all right, if we get to compare all the five C scores, we will get to know that here, okay, this is your highest C score of -0.39, which actually means that this model here, it signifies a high confidence level. Now the second one, if you can see here, the score which is of importance is your TM score. All right, so what exactly is the TM score? So a TM score is basically for measuring your structural similarity between the structures. The purpose of the TM score is to solve the problem of the RMSD, which is sensitive to the local error. So a TM score, if it is greater than 0.5, it indicates that the model is of correct topology, and a TM score which is less than 0.17, that means it is based on random similarity. All right. So that is so in this case, all right, the TM score, it shows a very good topology because it is greater than zero point 0.5, and that's why we can say this is a good TM score again. Now there is a third parameter, but in order to understand or see the third parameter, you have to click here, that is read more about C score. So I click on this, I go to the new tab. Yeah, so you can see the five models, you can see the five C scores, you can see the TM, you can see the RMSD. These are the decoys. You remember I was talking about you have these structural decoys, and these are the structural decoys which actually make it this ab initio based method. So whatever the model has been predicted is on this structural decoy proteins. How do you know the structure? How do we get to understand the structure decoy proteins? Obviously, from known PDB structures, this 600 structural decoys have been developed. Okay. So I was coming to the last parameter, which is your cluster density. So a cluster density is nothing but the number of structure decoys at the unit of a space. So a higher cluster density means that the structure occurs more often in the simulation trajectory, and therefore it signifies a better quality model. The values, if you see, okay, um, So basically, higher your cluster density, better is the confidence level. So if you see among these 5 models, your first model is giving us a cluster density of 0.249, which is higher than the rest of the models. Thus, by looking at your C-score, by looking at your TM score, and you're looking at your cluster density, we can actually come to the conclusion that model 1 is a very good predicted three-dimensional structure. So this is your model 1 structure. Now, if you want to download the model 1 structure, you simply click on this link here, okay, which says download model 1. And once you do that, the model will start to get downloaded. You can open the model 1 in PyMol, which is nothing but your structure visualization tool. This is your COVID-19 nucleoprotein, which has been modeled using Ab-Initio based method. So this is how we actually predict the protein structure using I-Tasser. The whole purpose of this video was to showcase the tutorial of I-Tasser for an unknown sequence such as COVID-19. Going into the intricacies of this nucleoprotein for COVID-19 because my major focus of this tutorial was to show you how to actually determine a or how to actually predict a three-dimensional structure of the protein if your if the sequence identity is less than 40%. And this can be done using this method which is called as an ab initio based method. So hopefully you understood how to predict the structure using I-Tasser. You can use this for your further research of COVID-19 sequences or any other sequence of any disease which you are actually working on. Hopefully you have understood this tutorial. Thank you. Hi, hope you liked the video. Do not forget to like and share this video. If you want to see more such videos, you can click here. If you want to see the videos which is related to this content, you can click on the playlist which is there below. And finally, do not forget to subscribe to this channel. Click.